The study group consisted of 75 children born to HBs antigen positive mothers who received vaccine and hepatitis B immunoglobulin after birth according to standard protocols. The first serological test showed no HBs antigen positive cases. This was confirmed by a second serological test from a different manufacturer. The real-time PCR showed that 21 out of 75, or 28%, were positive for HBV DNA. Following this, the standard PCR on individual HBV genes was carried out to amplify and to sequence the OBI-positive cases. Then, an extensive mutational analysis was undertaken on the samples, and in 10, or about 47% of patients, the viruses contain G145R mutation in the A determinant of surface proteins. Among OBI-positive cases, only 5 were positive for anti-HBc, and all contained protective levels of anti-HBs. Above the possible infection sources for post-vaccination hepatitis B occult infection, we think that, first, the blood of newborn babies contaminated with HBV particles in uterus during the delivery, and subsequently the HBV vaccine and HBIG were ineffective. Second, the A determinant variants found in the study may be responsible for occult infection and were undetected by conventional HBs antigen screening tests. Third, children infected with HBV mutants or simultaneously infected with the wild type and mutant HBV as a quasi-species. However, the detection of this population was not possible by sequencing analysis. Fourth, HBV mutations outside the surface protein may also be responsible for occult infection. 5. Low-level expression of HBs antigen due to low viral load that might just be enough for viral assembly, but is below the sensitivity level of standard tests, which making it undetectable. All patients in our study contained HBV viral load less than 10,000 copies per ml. 6. HBs antigen and anti-HBs formed complex, and as a consequence, the HBs antigen was hidden and could not be detected by ordinary serological assays. And finally, individuals protected by HBV vaccine had low immune response capacities at the early of childhood. As shown in our study, 52% of HBV DNA infected children had anti-HBs levels less than 100 international units. Above the reasons for such considerably higher number of OBI in our study, we believe that in almost all of the few investigations published so far, the prevalence of OBI ranged between 0 to 11%. Except of one study on African children, the rest of surveys have not included surface antigen negative high-risk children. In fact, authors selected surface antigen positive vaccinated children from either those born to HBs antigen positive mothers or those born to healthy mothers. Despite immunoprophylaxis in both groups, still a percentage of them contain mutations in the A determinant region. Secondly, the genotype of all infected persons was D. There are some hints in the literature that transmission efficiency of genotype D might be higher than some other genotypes. And the finding of absolute genotype D in our study and perhaps some host genetic and epigenetic factors could be responsible for such a high prevalence. I would like to conclude that we believe HBs antigen negative vaccinated children born to HBs antigen positive mothers do not represent as being HBV infection risk-free, and it is too early to reach to a definite conclusion about the real prevalence of OBI in vaccinated individuals, unless more similar studies come up in the future.